Very good morning, dear students. We have been discussing about unit number 6, the detailed aspects of lipids. So we have already discussed the simple lipids, that is triglycerides and waxes, compound lipids. There we have discussed phospholipids and glycolipids. Then the next one is derived lipids. And in the syllabus, one of the most popular derived lipids are steroids. So that is what we are going to discuss in this particular class. So before we are going to discuss steroids, you should know some basics of derived lipids. So derived lipids are the lipids which produced due to the hydrolysis of either simple or compound lipids. Okay, they are derived from simple or compound lipids. Sometimes there is a mixture of both, and they include fatty acid, glycerol, sphingosine, and steroid derivatives. So all those things we are familiar. Fatty acid, glycerol, and sphingosine we are familiar. And what are these steroid derivatives? So the steroid derivatives are phenanthrene structures. We will discuss what is this phenanthrene structure. Okay, so that are quite different from lipid made up of fatty acids. And this phenanthrene structure is the major attraction of this particular component. And the major types of derived lipids are steroids, terpenoids, and carotenoids. So these are three important topic. Lot of components, lot of contents are there to discuss. In your syllabus, they have mentioned the details about steroids. So thus we are going to discuss the detailed aspects of steroids. So what are steroids? So the name steroids, it is from stereos, that is solid. Okay, so they are one of the most studied classes of biological component, or are often found in association with the fat. So one of the most studied class of biological component that are often found in association with the fat. And the most important thing, they are not having the fatty acid and they are non-saponifiable. So what is mean by non-saponifiable? That means to say they cannot be hydrolyzed by heating with alkali to yield sop of their fatty acid component. You know that the glycerol is the principal component of fatty acid. That is the principal component in sop. Okay, so you know that. So they cannot be hydrolyzed by heating with alkali to yield sop of their fatty acid component. And in soap manufacturing, glycerol also will be produced as one of the important byproducts. And fatty acids are, but the fats are saponifiable and form soap when hydrolyzed with alkali. Okay, the saponifiable form of soap when hydrolyzed with alkali will give fats. All steroids are fused and fully saturated ring system that is known as the phenanthrene. That ring system is having a specific name that is little bit lengthy. In fact, that name is cyclopentanoperhydrophenanthrene or sterane, or simply call it as sterane nucleus. Okay, cyclopentanoperhydrophenanthrene is the major fused fully saturated ring system is present in the steroid. So all components, these basic components will be there. And what is this cyclopentanoperhydrophenanthrene or sterane? They consist of 3 cyclohexane ring that denote as A, B and C. They are fused in a non-linear manner, and there is a specific type of arrangement called a phenanthrene, where the D, the terminal D, that is a cyclopentane ring. There is 3 cyclohexane ring and there is a cyclopentane ring, and this manner is known as, this way is known as a phenanthrene manner. And this is what is the phenanthrene. Okay, so here you can see 6-membered ring 1, 6-membered ring 2, and this is not, if it is here then it is linear, but here it is like this. Okay, so that is known as non-linear. So you can see it is going like this line indicate, okay, the array, the order. Okay, so the direction, so that is known as a phenanthrene. And this phenanthrene in detail, this is as such, it is given here 3 6-membered ring A, B, C, and 1 cyclopentyne ring. What is that? 5-membered ring. And the arrangement of carbon is very interesting, the numbering. So it starts from here. Okay, carbon number 1, 2, 3, 4, 5. Then the next ring 6, 7, 8, 9, 10. Then the third ring 11, 12, 13, 14. Then these 3 6-membered rings are over. Then the next one 3 position 15, 16, 17. And this basic ring system is known as cyclopentanoperhydrophenanthrene or sterine nucleus, as I said. And so all sterine steroid molecule, this component is very common. And another important thing, in this sterine nucleus, carbon number 5, carbon number 10, carbon number 9, carbon number 8, okay, 5, 8, 9, 10, 13, 14. This many carbon atoms, that is these 6 carbon atoms are asymmetric carbon atom, okay, they are asymmetric carbon atom. So this carbon atom is having very important role in the biochemistry, the chiral carbon atom. Then these steroids are also can exist in the form of stereoisomers. Okay, so how the steroids will be existing, especially these 3 rings, okay, this phenanthrene ring, they are able to exist in the stereoisomers. So these 3 cyclohexane ring, on the basis of tetrahedral theory, okay, tetrahedral theory is capable of existing in 3-dimensional conformation, either of a chair or of a boat manner. Okay, a 3-dimensional conformation, either of a chair or boat manner. The chair form is rigid, and when it is changed to boat form, some angular deformity and invariability takes place. Okay, and this chair conformation of cyclohexane itself is more stable than boat conformation. Okay, the chair form, because you can see the groups are in the opposite side like this. This is a chair group. This is more stable than the boat form. Here you can see all the the ends are in the either side. Then in naturally occurring steroids, all the ring are chair form, and the ring can be either cis or a trans. Okay, and a ring A and B will be cis to each other, and this molecule is known as a normal configuration. If A and B are cis to each other, that is the normal configuration. But if A and B are the trans configuration, that is known as a allo configuration. Okay, so that is the important. They can exist in both cis form and trans form. Okay, cis form, that is known as a normal configuration. Trans form is known as a allo configuration. So this is the stereoisomerism commonly exhibited by steroids. And the stereoisomers of steroids, in the structural representation of steroids, there are substituents. If the substituents above the plane, that is the beta orientation, that is shown in a thick line. Suppose this substituents are like this thick line, this thick line, this thick line. That is these thick lines are above the plane, that is known as a solid line. Okay. Then the substituents that are below the plane of the ring, that is beta oriented, that is represented as broken lines. Okay, the substituents that are below the plane of the ring, then that is represented with the broken lines. Okay, that is the important, the stereoisomers of the steroids. Then Then the next one is the classification of steroids. Okay, the classification of steroids. Steroids are classified into 3 different groups: C29, C28 and T27. 27, 28, 29 steroids, 24 steroids, and 18, 19, 21 steroids. So these are the, that means to say the number of carbon atom is 21, 28, 27, one category. Number of carbon atom is 24, 21, 19 and 18, second category. Okay, and please note that in all the structure cyclopentanoperhydrophenandrine is common for, okay, common structure. And the classification of steroids, first one C29, 28 and 27 steroids. Example is cholesterol, corprostanol or corprosterol, cholestanol, ergosterol, lanosterol or cryptosterol, stigmasterol, that is commonly present in soybean and wheat germ oil, and spinasterol, that is commonly present in spinach, okay, or cabbage, okay, cystosterol, that is commonly present in the plants, okay, and ostriosterol, that is commonly present in the oyster, that is mushroom, okay, agaricus, pleurotus, okay, all those mushrooms, okay. Then chondrilla sterol, then marine sponges, okay, sponges, they are obtained from the marine ecosystem, okay. These are the 27, 28 and 29 sterols. Then what about C24 sterols? They are bile acids, okay, and 18, 19, 21 sterols are, if it is 21 sterol, digitoxior or under digitalin, then quercetin, then progesterone, okay, then C90, that is testosterone, and 18 is the estradiol, okay. These are the major types of commonly studied steroids. Then the first one is cholesterol. So this is the most well studied component in the some cell membranes and in plasma lipoprotein, and they act as the precursors of many other biologically important steroids such as bile acid and other steroid hormones, okay. They are very, very important. So it is the principal steroid of higher animals and is especially abundant in the nerve tissue and the gallstone, okay. And it is first isolated in 1784 from human gallstone, which consists of almost entirely of cholesterol, okay. Its main source are fish liver oils and the brain spinal cord cattle, okay. Then the white matter contains 14%, grey matter contains 5%, spinal cord contains 21%, and liver contains approximately 1% of cholesterol, okay. The cholesterol is not commonly found in plants, and the structures of cholesterol was determined by a German chemist known as Adolf Windaus, okay. Adolf Windaus, he is the person who studied the structure. And cholesterol, the molecular formula is C27H45OH, and in addition to hydroxyl group at the C3 position, there is a double bond at the C5 position. We will discuss that structure. And the hydroxyl group constitute its polar head, and the remaining molecule is hydrophobic, and it is a white crystalline solid, and the optical activity is, okay, that is 39 degrees degree, and it has a high melting point, 149 degrees centigrade. And a great important thing, cholesterol, the study of cholesterol, the biosynthesis of cholesterol, the biosynthetic pathway of cholesterol, that a scientist called Kannur Bloch received a Nobel Prize in chemistry, okay, in chemistry, and that is a high blood cholesterol results in the deposition of cholesterol in the arteries, okay. So high blood cholesterol, if it is deposited in the arteries, that leads to the atherosclerosis, that you know actually, okay. Then the aerial surface of the roughness due to the cholesterol deposition, that leads to a specific condition called thrombosis, okay. These are the some of the deleterious aspects due to the accumulation of the cholesterol, okay. This is very, very important. So this is what the structure, as I said, okay. So this is the simplest structure. First we will go this one. This is the cyclopentanoperhydrophenandrine A, B, C, D, and here you can see this is the structure. Okay, and here there is a hydroxyl group. This is a methyl group. Okay, so here you can see 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And please note that between 5 and 6 there is a double bond. This is what I told. It's very, very important. Okay, and there is 19th position, there is a methyl group. Okay, then the 10, 11, 12, 13, 14, then 15, 16, 17. Okay, here you can see another methyl group, then 20th, then CH3, CH2, 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 CH, CH3, CH3. Okay, so this is the 27 component. That is what I told you, this particular component. So this is the detailed structure, okay, of cholesterol. That can be simply we can return. This can be simply we can return like this. Okay, this is the structure of cholesterol. Then the other steroids are coprostanol. The coprostanol occurs in the fecal material and is produced in the intestinal as a result of bacterial action in the double bond of cholesterol. Okay, so here you can see double bond of cholesterol will be acted by the bacterial enzyme and that will be converted into coprostanol. Then it has an AB junction is cis coprostanol. Okay, the A and B junction, they can act as a cis form, and the lanosterol is a C30 compound with the twin methyl group at C14 and third angular methyl group on the C14 component. Okay, so this is the structure of ergosterol, the basic 6-membered cyclopentahydrophenandrine ring, the hydroxyl group, this one. Okay, and this 23 and 28. Okay, all those things, and you can see the hydroxyl group. Okay, the methyl group. Okay, here also you can, this is lanosterol. Okay, so a little bit almost structures are the similar. Okay, only difference is here there is a methyl group. You can see the remaining everything is the same. Then these 24 steroids or bile acid. The bile acids are important end products of cholesterol metabolism in higher plants. Okay, all these are derived from the C24 parent steroid called cholinic acid. Okay, and the most abundant bile acid in human are cholic acid, approximately 25 to 60% of the total bile acid. That is another derivative is quinodeoxycholic acid. Next one is deoxycholic acid. Okay, this is 25 to 60 and this is 30 to 50 and this one is 5 to 25%. Okay, and the various bile acid offers from each other in the number and position of hydroxyl group. That is the major difference. Okay, here you can see cholic acid, basic phenandrine ring A, B, C, D. Then here is hydroxyl group, carboxylic acid group, there is hydroxyl group, there is hydroxyl group, there is hydroxyl. Position 3, 7 and 12 there is hydroxyl group and there is a carboxylic acid group. And what is the difference between this one? You can see almost the same. Okay, and that lack this hydroxyl group is absent here. That is known as quinodeoxycholic acid. This is cholic acid. Then C21 and C19 and C18 steroids, they are mainly hormones. Okay, several important poisons are based on these particular structures, and another important components are digitoxor digitalin. It is present in the foxglove plant. Okay, that is commonly present in foxglove plant, that is digitalis purpura, and has a very powerful, these components act as a powerful heart stimulating acting component. Okay, then another component is known as queabine. Okay, that is obtained from an East African queabia tree. Okay, and has a sugar rhamnose and attached to sterol nucleus. Okay, and here you can see the cardiac glycoside also present here. Okay, cardiac, like we said, cardiac glycoside. And hormones, you know that is progesterone. It's a hormone acting on corpus luteum. Then deoxycortisone produced by adrenal cortex, hormone again. This is a molecular formula. And cortisol, again adrenal cortex, is again a hormone. These three are C21. Next one is C19. Okay, there's testosterone, that is testis. Okay, then that is hormone again. Then C18, that is estradiol, that is ovaries, and hormone, male hormone and female hormone. Okay, this progesterone is a pregnancy related hormone. Okay, so these are the important steroids. So this is all about the steroids, and today we can conclude. Okay, so we can glance once again what are the major points. We have discussed the introduction to derived lipids. Then there are three types: steroids, terpenoids and carotenoids. But we are discussing in detail about steroids, structure of steroid nucleus. We have discussed the stereoisomers, the both chain and chair form and boat form. Then the classification of steroids we have discussed. Then there are three types: C29, 28, 27, 24 steroids. Then 29, 19, 18 steroids. Then we have discussed the examples. Cholesterol comes under this family. Okay, and other steroids are like in this particular first class, that is propranolol and cholestanol, ergosterol and lanosterol. And C24 steroid, that is bile acid. Then C21, 19 and 18 steroids, that is quercetin and digitoxyr and digitalin and all other things in addition to the hormones such as progesterone, testosterone, estradiol, etc. Okay, these are the things we have discussed. So these are the major textbook and reference book, dear students. These are the major book where I always highlight Leninger Principles of Biochemistry and Biochemistry by J.L. Jain. Thank you very much for watching this particular class. Okay, I know there are little bit of sound. I'm sorry because there is torrential rain that is happening while I'm recording this particular video. Okay, thank you very much.